म्हणून म्हटलं.
सर तुमचा आवाज ब्रेक होतोय खूप. हॅलो सर.
हॅलो. शेअर करू ना सर? सर शेअर करू का YouTube वरचा?
हो करा करा करा.
आणि सगळ्या पार्टिसिपंटला पण पाठवतो.
मी करू ऍडमिट यांना बाकीचे?
हो करा तुम्ही ऍडमिट. सर गांधी सर म्हणतात शेअर द झूम मीटिंग.
काल केलं ना आपण, याला ग्रुपला टाकलं ना का? तुम्ही केमिस्ट्रीच्याच ग्रुपला टाकलंय फक्त?
केमिस्ट्रीच्याच ग्रुपला टाकलं ना झूमचं. सरांना तुम्ही पाठवा. सगळ्या ग्रुपला नको, फक्त
ठीक आहे.
गांधी सरांनाच पाठवू ना? पाठवलं.
सर्व ग्रुपला पाठवा सर.
हो.
Apart from chemistry, you can add all groups. इवन केमिस्ट्रीला पण पाठवायला हरकत नाही. सपोज काही जण नाही अटॅच झाले तर दे कॅन युटिलाइज. ओके,
नो प्रॉब्लेम. येस सर. येस.
यस.
सर YouTube केलं स्टार्ट?
हो केलंय. येस. हां. हॅलो. हॅव स्टार्टेड सर ग्रुप YouTube लिंक. हॅलो.
हॅलो सर बोला.
हां येतोय ना आवाज?
होय 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 चालू आहे.
Mahaba ma'am, just ask to Mallika ma'am.
Yeah, tell me Durgesh.
Yeah, ma'am.
Ma'am, you are there now?
Yeah, I'm here.
Yeah, okay ma'am.
सर एक चॅट बॉक्स मध्ये रिक्वेस्ट टाका की ज्यांनी मोबाईल जसे हे केलेत ना त्यांनी रिनेम करा असं.
काय करायचं? रिनेम ना?
All the participants please rename themselves with their name.
Oh yes, started. Hello. रुकारी सर,
हा सर बोला.
जस्ट सी बघा ते YouTube is working or not. Total
YouTube
39 participants are joined right now.
चालू आहे सर YouTube. 33 वॉचिंग ऑन YouTube.
सर इथं 27 आहेत ना ह्याला झूमला. 27 दिसतात मला टोटल.
हो 27 सध्या आहेत सर. अजून ऍडमिट करतो मी परत. 13 जण वेटिंग वरती आहेत.
वेटिंग वर जे ते ऍड करा.
हो केले सर.
हॅलो.
YouTube 39.
Total 45. Hello, principal sir. Hello. Hello. Hello.
Durgesh sir.
Yes sir. Shall we proceed for right now 45 participants are there sir.
Better ask ma'am, Mallika ma'am, okay?
On YouTube 45 are
Shall
there.
we wait for some more?
Yes sir, 5 minutes.
Okay, minute, wait.
रुकारी सर.
हा सर.
मी काय म्हणतो समजा जर मी इथून मला कारण की ते सांगू दोन्ही
नाही मी बाकीचं तुम्ही हे सोडा. मी ऍडमिट करतो.
नाही युट्यूबचं मी इकडून बंद करून ठेवू का म्हणजे मग तुम्ही मला
हो.
दोन्ही आवाज येतात.
हो YouTubeचा तुम्ही म्यूट करा ना खालून.
म्यूट करतो. हा
म्यूट करा.
केलं.
काय प्रॉब्लेम नाही.
ओके. Right now sir 45 participants are there.
YouTubeला आवाज नाही येते वाटतं. तुम्हाला येतोय?
नाही येतं सर आवाज. ते आपण
येतो
जे
ना?
बॅक ते सगळं रिपीट होतंय. माझं तिथे चार्जर आहे.
महाबळ मॅडम अटेंडन्स झाली ना रेडी?
येस.
हाय सर. लिंक सगळ्यात शेवटी लास्ट 15 मिनिट्स मध्ये शेअर करणार मी व्हॉट्सअप ग्रुपला.
ओके चालेल.
कारण आधीपासून शेअर केली तर मग ते अटेंडन्स भरण्यातच लोक वेळ काढतील. इकडे सेमिनार मध्ये लक्ष नसेल.
Hello.
Hello.
Yes, ma'am.
Yeah, good
Right now,
morning.
ma'am, 45 participants are there. I think we should wait for 5 minutes and then we'll be proceed.
Yeah, it's fine. No issue.
Okay, ma'am. No.
YouTube 85.
85. Okay, good, sir. Sir, hello. Dukari, sir. Hello. Hello.
हा
But
सर
ma'am,
बोला.
audio is not clear. Said by Mallika, ma'am. Audio is not clear.
कुठे?
It's okay.
Mallika, ma'am, saying audio is not clear.
Where audio is not clear?
I
Yeah,
think from ma'am side. Ma'am side.
अच्छा, ma'am side. Okay.
Yes, sir.
it was disturbing actually.
Yes, ma'am. Some participants are joining, ma'am, right now.
Okay.
That's why some hurdle is there.
No issue.
Okay, ma'am.
महाबळ मॅडम.
Yes, sir.
Might be background music.
एक मिनिट सर. फॅनचा नॉईज येतोय का तुम्हाला?
No, no.
कारण नाही थांबा, माईक वरती इथे बघितलं ना
Hmm.
तो बॅकग्राऊंडचा आवाज थोडासा हे येतोय.
एक मिनिट हा, एक मिनिट.
मॅडम तुम्ही म्यूट करा बरं.
मी म्यूट करू का? एक मिनिट.
हो. आय थिंक डोअर जर क्लोज करता आलं तुम्हाला तर म्हणजे हे होईल, डिस्टर्बन्स थोडा कमी होईल थोडा.
Sir,
बोला.
what about that snipping
Yes, sir.
tool?
Sir, we can proceed now.
Proceed na, sir? Okay. No problem.
Yeah.
Sir.
ओके, मी करतो, सर पण करतील, काय प्रॉब्लेम
Okay,
नाही.
okay. Malvika ma'am, Dr. Malvika ma'am?
Yeah.
Yes. Okay, ma'am. Shall we proceed, ma'am?
Yeah, we can.
Okay. Yes. Good morning to one and all the participants, PG students and faculty members from throughout the different parts of our country. On the behalf of Yashwantrao Bhosle College of Pharmacy, Savantwadi, I, Mr. Durgesh Toliram Gautam, Assistant Professor and Program Coordinator, I welcome you all for this 3 days live webinar series of Technical Lecture Series 2020. Today, we are having a session of Dr. Mallika ma'am on machine lecture in drug discovery. So now before proceeding for today's session, now I would like to request our principal, Dr. V. A. Jagtap sir, to give a welcome address to all the participants.
Good morning all. Hope all you are fine and healthy. We know that we are going through pandemic crisis, but positively we are attached and connected with each other. In this lockdown period, Yashwantrao Bhosle College of Pharmacy has organized various online activities through which they have conducted various online quizzes, some surveys, particularly training courses and webinars. So extending these activities, herewith we are presenting especially live webinars of technical lecture series for informed students and faculty. So I'd like to welcome you all, and I'd like to welcome today's honorable speaker, Mallika ma'am. She has accepted our request for today's session with her kind gesture and enthusiasm. Her vast experience will give you a glimpse on today's topic, but I will appeal you all to be connected with her through her Mass Academy, Hyderabad. Welcome, ma'am.
Hello.
I'd like to thanks also to the all faculty members of Yashwantrao Bhosle College of Pharmacy, as well as coordinator, session coordinator, Ms. Rashmi Mahabale madam, as well as webinar coordinator, Mr. Durgesh Gautam sir. And especially I would like to thank Mr. Tushar Rukari sir also for arranging all these technical activities. I wish all the success for today's topic. Welcome you all. Thank you.
Okay, thank you so much, sir. Now, I would like to request Ms. Rasmi Mahabad ma'am, Session Coordinator and Head of Pharmaceutical Chemistry Department. Please proceed further. Over to you, ma'am.
Good morning to one and all. Beloved Principal Dr. Vijay Jagtap sir, convener of this webinar series, webinar coordinator Durgesh Gautam sir, along with Principal Tushar Rukari sir, and today's speaker Dr. Mallika. Along with all the participants, I welcome you all on our series. It's my privilege to introduce today's speaker, Dr. Mogipalli Mallika, that is Mallika Alwala, ex-professor of Sniper Hyderabad, and she is also working as founder and instructor at Mars Training Academy. It's really a proud moment to introduce you, ma'am, over here. She has more than 10 years of experience in drug discovery programs involving in the fields of tuberculosis, cancer, and metabolic disorders. She is expertise in handling drug design software like molecular modeling software, Schrodinger, Autodoc, Silbil, and many more. She is expertise in high throughput screening, assays of identification of novel small molecules inhibitors, including CERT, HDAs, and corinstase mutase, like many more molecules she has handled. She is also expert in quantum mechanics, simulations, SBDD, LBDD, ADMET studies using in silico tools. And also she has worked in cell culture and various animal models. She's working as a team member in designing research strategies. Also, she is involved in the number of practical sessions of computer lab basics, database retrieving, introduction to Linux system molecular modeling software packages, a number of grants she has fetched from DBT for cancer biology, then DST, ISAR Pharma Private Limited. She has 53 research publications on her name. She has played a role as a speaker, resource person for many international national conferences as well as QIP. Also, she has done the job of evaluator for poster competitions. Awards in her name are Associate Fellows of Telangana Academy of Sciences for 2017. She is also received award of Best Research Scholar of the Year 2012 from Pharmacy Department, BITS Pilani, Hyderabad, which is sponsored by Bharat Biotech, Hyderabad. And she has also won a first prize in poster presentation in APTI National Level Conference held at Hyderabad. So with this small introduction, I would like to request ma'am, Dr. Mallika ma'am, to proceed with her session. Over to you, ma'am.
Yeah,
Yes.
thank you, Rashmi. Good morning to all, and thank you, Dr. Vijay Jagtap and Rashmi, to give me this opportunity as a speaker. And I also thank Durgesh connecting me via Zoom without doing any hesitation. And so today I would like to start my session with the motto of giving something to the students as well as faculty, and the glimpse of machine learning, which will be emerging field in nowadays. So in order to avoid the bandwidth, I'm going to just close the video session and only the audio will be available for you people. Yeah, Dr. Durgesh, can you please give me share screening
Yes,
option?
Yes.
It was disabled.
Just a minute, ma'am. Share the screen.
On the right side of my window, it is available. You can just give the make as a
Okay, just a minute, ma'am. Allowed makers. Yes, you can start, ma'am.
But it is giving me message like host disabled the participant screen sharing.
Okay. Rukari sir, we can proceed for the as a make a host to Dr. Mallika ma'am.
No, sir. Wait.
Just a minute. Screen sharing is not there.
Madam, please try now.
Yes, yeah. Thank you. Hope is it visible to everyone? My
Yes, ma'am.
presentation.
Visible.
Yes, ma'am. Yes, we can see.
Yeah. Today, I'm here to present a session on machine learning in drug discovery. So all of you know about drug discovery as well as machine learning. Like it was very emerging field. So, but today the aim of my presentation was to give some basics and the flavor of machine learning so that everybody, whether they are a programmer or a chemist or a pharmacist, would like to learn the machine learning as well as programming. So I'd like to start my session always wherever I'll go for presentation. I starts my presentation with a quote given by the chemistry Nobel laureates in 1998, stating that in the real world, this could eventually mean that most chemical experiments are conducted inside the silicon of chips instead of the glassware of laboratories. So turn off that Bunsen burner, it will not be wanted in 10 years. So this was a great statement I like. So wherever I'll go, I used to start with the session with this quote. So hope you understand the importance of in silico drug design. The scientists have predicted in 1998 itself. So going to the drug discovery process. So this was very like thinking picture, like what I have to do in drug discovery. So when you start your career in pharma, industry or academia, you will be thinking what I have to do, where I have to start, and where I can reach. So these are the queries will come into either a student or a faculty when they start their career. So we will be thinking that we are going to do something to the society. So with the aim of that one, the people will struggle to study academics, then finish their academics and they go back to the industry. When they reach to the industry, the important skills which are required are nothing but your practical skills. So practical skills as well as theory skills are very important when you want to flourish in the industry or anywhere. So this was the picture which is not related to that. I want to just tell you, in a drug discovery process where the traditional drug discovery, people when they have started initiating or finding out the antibiotics like penicillins or paracetamol or cox2 inhibitors, anything, they all molecules has come into the market in a traditional way, not by using any in silico methods nor by any machine learning methods, but still we are using paracetamol is one of the best drug for the fever. So I'm not saying that traditional method of synthesis is not good, it is very good, but there the luck plays a major role. So which was basically depending upon the screening method. People used to synthesize a lot of molecules and they try to screen on various types of models. Based on that models, wherever it is giving the activity, that activity will be claimed as a one of the drug of choice for particular disease condition. So if you think of a traditional way of designing, it is based upon the random and trial error method based, and it will be taking more time consuming because you have to perform lot of experiments to understand on which disease condition your molecule is going to give the activity. So when you are repeating the experiments for many of the activities, the process will be expensive and laborious, and the success rate was very low when compared to the today's scenario. So from the past 2 decades, the CAD came into the picture. So people try to understand what is the structure of the protein and how the molecules looks like, how they behaves. So the biology part has been improved a lot. The success rate in the biology understanding was increased. As the success rate increased in the biology, the chemist could able to understand what is the protein structures and what is the nature, where it is located, how it is going to be effective for the disease condition. So by understanding the biology of the disease status, the chemist and other scientists like formulation scientists are trying to generate good number of molecules which can be showing potential activity. So here in the CAD scenario, the synthesis of the molecules or the designing of the molecules basically depending upon the reason. So so before starting any synthesis, we will have some questions like why I'm synthesizing, what for I am synthesizing, in which target it is going to be acting, what are the off targets of my molecule. So all these questions will be raised before you start designing a molecules by using CAD. So it was target specific and structure based. As we are using computers, the process has become fast and automatic, but here the logical thinking will be coming from human brain only, not from the computers. So the human brain has to work along with the computers to get a good results. Because of computational facility, you are not going to repeat all the experiments. You are going to calculate ADME properties and some of the log P properties, minimum properties by using computational tools. Hence, the cost has been tremendously reduced and the success rate has been increased. So here the success rate means not only for the particular target protein. If you are designing any molecule for particular protein, accidentally, if they are showing good affinity towards the other protein, the people are diverting the molecule to repurposing to other proteins or other disease conditions. So the success rate has been increased by using the CAD when compared to the traditional methods. Now the present scenario was artificial intelligence and machine learning. So in machine learning, we are using computers. Durgesh, can you make other participants mute? So the artificial intelligence and machine learning era has come, where the computers can able to take the decisions. So how the computers can take a decisions is the question. So before the computers will take a decision, the, the programmer or the human has to write a programming, so that every time the intervention or the association of humans is not necessary. Once you write a programming, the computers are going to by default take a decisions based upon the instructions you have provided in the programming. That is what now the era is going on, machine learning, which enables a system to learn from the data. So you are going to give the data. We will see in coming slides which forms of data we are going to give it to the computers. So you have to give a lot of data to the computers, and the relation between these data will be automatically done by the computers. Once that is done, the output is going to come based on either whatever you have provided as an output has to come, or it may give new ideas or new correlations. From that correlation, you can able to understand what was the connectivity between the disease condition or the drug molecule or the protein and a drug molecule on various parameters we are going to see now. So as once you are going to write a program, and every time it's not required to your assistance for doing the calculations, it is going to be very low cost, and it is very fast because the computers can read almost thousands of publications in fraction of seconds. But if you want to read, it will take minimum 10 days to read the publications. So the connectivity, the machine learning and artificial intelligence mainly helping the scientists to understand what is already existing data is available in the literature, because from past 40 years we have doing so much of experiments and clinical trials and generating lot of data. So the data has been present in the databases, and there is no like connectivity or the understanding between the parameters has been done till now. So as the machine learning came into the picture, the people are trying to understand the connectivity between the parameters which are already experimentally observed, instead of designing new molecules and doing the experiment extra. So but the important thing here is you need little programming knowledge. Apart from your theory knowledge about the biology, chemistry, and pharmacy, you need some of the programming languages. Though it is not your core subject, you have to learn. So I would like to start the session with the drug discovery process and development process. So as all of you know that it was very long process. The drug molecule coming into the market means it takes long time, and also the lot of money will be involved. And so the discovery process includes identification of the disease. So this identification of the disease will be done either by pharmacist or the doctor. Why? Because the doctors knows very well if any new disease condition is appeared in any of the patient. They will try to understand what is the etiology of the disease condition, what are the parameters they are going to be changing, how it is important for the human population. Like the COVID has been came into the picture. Every day we are seeing new symptoms, like it may be having blisters, or it may be having ear infection or eye infection. Every day one new symptom is going to be identifying. So based upon the symptoms and the severity of the condition, how it is affecting the population, the disease condition is going to be identified, whether it is important to control that disease, or it is not going to be that much important to design a molecule fastly. So once you feel that the disease condition is very important to handle, then people are trying to isolate the proteins which are involving in that particular disease condition. Here the biologist comes into the picture, or the pharmacologist can come into the picture. They will isolate the proteins, and they will understand what is the upstream or downstream proteins which are connected with that particular protein, and how this protein is going to be like having three-dimensional structure. So all those crystallography studies and biology studies are going to be involved in the stage 2, where you are going to isolate the proteins. Then after that, once you feel that that protein is very important for the pathogenesis of the disease condition, then it is as a pharmacist or a chemist, we have to design a molecule which can binds with that particular protein and combat the disease condition. Once that is done, the preclinical stage is coming into picture, where you have varieties of uh, animal models you have to do, normal animal models and diseased animal models, and you have to understand the acute and chronic toxicity levels. Once the preclinical testing has been done, the formulation scientist will come into the picture, where he is going to design the molecules into a proper formulation. So that formulation is going to reach to the population. So the next important thing was the coming to the informatics role, because this is a discovery process which was following from olden days onwards. Now also we are following the same thing, but the speed up of the process has been done because of the presence of informatics. Informatics is nothing but the databases which are available, and you have a lot of tools by which you can do the calculations to understand the use of different tools to identify the disease conditions and potential targets. So identification of disease condition can be done by understanding the statistics of the WHO, what is the status of the disease condition, how many population are getting affected, whether globally or nationwide. Based upon that, it is going to be identified about the severity of the disease condition. Then the identification of the potential targets, that is equal to isolation of the proteins. Instead of isolating the proteins and designing the 3D crystal structures and upstream and downstream pathways, you have a lot of tools are available which can identify the druggability of the molecule proteins. Druggability is nothing but the capacity of the protein to be targetable, like pathogenicity of the protein molecule. So here you are going to calculate the druggability score by using the tools. And the three-dimensional structures, it's not compulsory to have a crystal coordinates. You can have still homology models by which you can utilize the structures. So once the structures has been identified, designing of small molecules, inhibitors are the activators. I can call it as a modulators of that particular protein. So this designing has been enormously increased because of the improvement in the computational tools. So we have like structure-based drug design, ligand-based drug design, and lot of varieties of strategies people are following to understand the binding pockets of the protein and the physicochemical properties of the protein binding site. Based upon that physicochemical properties, the people have started synthesizing the new set of molecules. So before they are going into the another preclinical and clinical stages, people are trying to understand what is the ADME properties of the molecules and pharmacokinetic properties, metabolism properties. Everything will be in silico, you can predict. Then coming to the formulation point of view, because that is very critical step. If you want to bring a molecule into the market, you have to understand which form of the molecule can be stable and which can available 100% bioavailability in the bloodstream. So you require combinations of the polymers which can be added to the drug molecule. So in case of formulation optimization, you have the in silico studies are available, where you can understand the polymorphism of the polymers, interactions, and the stability studies. So once discovery stage over, the molecule enters into the clinical trials that takes around 2 to 10 years. Then it will be going to the FDA approval and comes into the market. That is the developmental stage. So in discovery stage, we have seen in all the varieties, we have informatics tools. Similarly, in developmental stage, we have big data analytics and machine learnings are available, which will be helping to understand the data which is provided by the clinical trials, and also post-market surveillance data. Everything will be captured by using the various statistical packages, as well as the e-clinical and Oracle clinical remote data capturing methods. So this is very important stage where you will understand the correlation between the clinical trials data. So this is simply how the drug discovery process is going and how this informatics is playing a role. This is which is related to the CAD. So coming to the next improvement, that is artificial intelligence, machine learning, and deep learning. So nowadays we are seeing everywhere these three words are emerging. So we would understand what is the difference between these three methods first of all. So the artificial intelligence is a method by which humans are going to simulate or write a program in such a way that the machines can simulate the human behavior and take a decisions. See, you have nowadays Google capturing, which you call it as, if you see the face of the person, Google can easily say that there are two eyes, one ear, two ears, and one nose and one mouth, so that it can identify and says that it is a face. So the capturing of the features can be done, and the machine can directly take a decision. It is corresponding to the image of the face. So that type of simulation will be called as artificial intelligence. So this is a broader way. But this artificial intelligence cannot tell you whether this face is corresponding to the human being or this face is corresponding to the some other animal, because animals will also have same type of features, two eyes, two ears, and one nose and one mouth. So but artificial intelligence will help you to give a clear idea quickly what is the image corresponds to. Coming to the machine learning, it is nothing but the ability of the machines to automatically learn without being explicitly programmed. So in machine learning, what you are going to do was, by using a certain set of molecules or the data, you will design some parameters. And when you give new set of molecules from the already existing data set, it try to correlate the properties and gives you the outputs. That is your machine learning. We will see in next slides very easily. Then the next method was deep learning. Here, the system itself will understand the connectivity between the parameters as well as it gives the decisions. So when you are doing or leaving all the decisions and the correlations to the machine itself, then it is called as a deep learning. But if you are providing some inputs and asking the output, then you will call it as a machine learning. So coming to the difference between machine learning and deep learning. So in machine learning, this is the output which I am giving. You can see the input image. It's not a numerical value. It's not some alphabetical text. Here the input in the form of image. You can understand that image is corresponding to some tumor size which was taken by CT scanning. Okay. So here we are going to give the input and trying to give some features. What are the features of that image? What is the size of the tumor? What is the texture of the tumor? What is the length, width, then weight of the tumor? Then once you do the CT scanning, layer wise scanning can be done. Then it is going to tell you how many different varieties of patterns are present in the tumor. So all those features you are going to define. Once you define the features like weight, size, then texture, I'm taking only 3 examples over here. And you are allowing the system or the computers to understand the correlation between these 3 features. So here I have given only one input. You are going to give almost 10 to 20 inputs, which are of a different different CT scans of the 10 different patients, for example. So once you give the features, then it is going to extract that features and trying to understand what is the connectivity between these 3 features: weight, size, and the texture. So based upon that, it is going to tell you whether this is a benign tumor or a malignant tumor. So how the system is going to identify this is a benign or malignant tumor? When you are providing the features to the system, you will say that if the weight was 10 mg and the texture was smoothie texture, then you are going to say that it may be benign tumor, because the scientist or the doctor, they knows the parameters by which they can identify the benign tumors. So if the weight was more and the blood vessel count was very high, then you are going to define it as a malignant tumor. So based upon the features which has been connected from the classification, the system is going to tell you whether it is a benign or malignant. Similarly, in case of deep learning, you are going to give the inputs as a tumors images. Then you are not defining any features. The system itself identifies the features. Based upon the features, it will try to understand the connectivity between those features and gives you whether it is a benign or malignant tumor. So in case of machine learning, you have to do two works. One is giving the inputs and defining the features and what is the output you are expecting. But in case of deep learning, you have to give only inputs and remaining things system is going to give you the outputs. So the data which you want to give a input to the machine learning or deep learning can be of varieties of types. You can give the data in the form of tables or numbers, images, videos, text, graphs, and click streams, as well as mixed data. You can give some of the images and some of the graphs, and it can understand. And click streams are nothing but the paths, how many times you have opened your website or how many times you are going into your particular URL site. So all those click streams can also given as a input data. So any type of data can be understood by the machine learning. So coming to the classification of the machine learning, as I said you, the machine learning program tries to get a connectivity between the given features. There are three varieties of machine learning models are available. One is supervised learning, unsupervised learning, reinforcement. In supervised learning, it was basically task driven. So because you are going to define input as well as features, the supervised learning always try to understand the connectivity between the given features and what is the output you have defined. And this supervised learning can be performed by using different methods. One is a regression based method, decision tree based method, random forest method, then classification based methods. So here two types of data we can give. One is continuous data, other is categorical data. So continuous data means you have a data which is linear in ratio. Like if you take absorbance versus concentration, the always you will see there is a correlation between the absorbance versus concentration of a assay. So that type of data if you are using, then you can use regression models, decision tree and random forest methods, which can be called as a continuous data models. If you have a categorical data, there is no linearity between the x-axis and y-axis data, but there is a logical connectivity is there between two parameters. In that case, you are going to use a classification based methods. So in regression methods, we have linear and polynomial, as most of you knowing simple linear regression and multiple linear regression models. So those are nothing but same here. So you are going to use statistical MLR and SLR methods to do the supervised machine learning programming. Similarly, unsupervised machine learning, where the data driven. So here you are not going to give any features. The program itself will understand and extract the features as well as the connectivity. So it is basically depending upon the data driven, not on a task driven. So whatever the data you are giving, it will try to understand that data and gives the output in the form of clusters, not in the form of benign tumor or malignant tumor. It says that there are features which are common in some set of molecules and there are features which are common in another set of molecules. Like that, it is going to segregate your data and gives the outputs. So if you have a continuous data in unsupervised learning, you will use principal component analysis and k-means and SVD methods. Similarly, in categorical methods, you are using the association analysis and hidden Markov models. These are the different varieties of the statistical methods will be applied by using unsupervised machine learning. Then you have a third method, which is reinforcement. So in this method, basically the algorithm learns to react on environment. So it is a trial and error method. So it is going to understand the connectivity and the feature extraction. And whenever you give a text set or validation for the any of the molecule for the text set, it will try to understand whether the data which you have been given is correct or not. Based upon that, it will go back to the initial programming and modifies the feature selections. That is reinforcement method. It is nothing but trial and error based method. So these are the three varieties of machine learning methods. So whoever will do the machine learning, your story will be revolving around these three varieties of classifications. So based upon the data which you are providing, you have to decide whether you have to do a linear regression analysis or logical regression analysis. So a system cannot understand which type of program it has to be used for the calculations. You have to tell if there is a linear connection between the concentration versus the absorbance, then you have to use a linear regression method. If there is no connectivity between the concentration and the absorbance, then you have to use logistical regression where there is no linear connectivity, but there are some ups and downs are there in the relation between these two parameters. So you have to, based upon the data, you have to decide which type of learning program you have to implement for the data set. So coming to the supervised learning, I'll just give some examples so that you can easily understand. So if you ask some school going guy, like how you can differentiate apple and banana or any orange fruit. So first of all, if the boy is very small or kid is very small, they can capture the images. They cannot understand the taste and everything. The colors are going to be observed by the kid and that could be captured. So whenever you ask any 1-year-old kid what is apple means, whenever he sees a red color one and which is a round like a shape, then he says it may be apple. Okay. Similarly, if it is a round shape and yellowish green combination of color, then he says orange. Then whenever he sees a curve shape of fruit and in a yellow color, it can be captured as a banana. The name has not been given by the kid. By seeing the colors, he can imagine that one. So similarly, the supervised learning, when you give the data, you are going to define the parameters. Like if it is going to be apple means the parameters like round, smooth and red will be already given as a features. Similarly for orange and banana. So what the system will do? So these two steps are the like inputs and the features, which we have discussed in the previous slide. So these two are going to be present are given by the programmer. And here you can understand the output also I have defined like round, smooth and red color. The output should be apple. So the system is going to make a decision tree. So decision tree is nothing but the classifier, which we have seen in the previous class. So in this, first it was asking a question. So this decision tree will be created based upon the features we have mentioned in the feature selection. First, the question was raises, it is round. If it is S, it is going to ask whether it is an orange or a apple. So here the another query has to come, that is whether what is the shape, whether it is smooth or bumpy. Okay. If it is S, it is asking whether it is apple or orange. If it is no, directly it gives the output as a banana because the first feature was round, which is not satisfied with the banana. So once the it is shape may be round shape. Instead of orange, I have to put the query as it is round. If it is round, then it is going to ask yes or no. If it is yes, again it is going to give you two answers. One is orange and apple. Then what is the texture of the fruit? If it is smooth, then you get output as an apple. If it is bumpy, you are going to get it as an orange. Like this, the system try to generate its own decision trees based upon the features which you have defined in the input. So this all these things can be done in supervised learning. Now I am going to give a new figure as my text, or I want to just test my program whether it is able to identify the banana as a banana or not. Now you can see the color is something in red color, but the shape was curved. So the first query was it is round. When it says the first query itself, though it is red color, you are getting output as a banana because you have defined the first query was the shape of the fruit. So like this, supervised learning try to generate the connectivities between the features and gives the output. It's very simple to understand. So this was with related to fruits, but you can see here the data which we have seen in the previous class, like the images of the CT scans of the cancer patient will be taken, and their images like screenshots or the slices of the images will be captured, and they are going to give the training data. And based upon this training data, whenever you give a new image or new CT scan image, it will try to overlap on these features and gives you whether it is a benign tumor or a malignant tumor. So this is very simple. This is called supervised learning. You are going to supervise the machines with the features, and the job which can be created by the machines was making a classifier or the decision tree. So coming to the unsupervised learning. So there are some set of animals which are present, and you're asking a small kid, what are those? Because we are not defining anything like this is dog or this is cat. We are just saying there are 5 animals are there. What is that? Then the kid is try to understand by the capturing the images. Like he can understand there are 3 very big animals and there are 2 small animals in a first go. So tries to understand what are the different varieties of data is present. So it try to make a clusters and association discovery. So when you ask the kid, he says generally that there are 3 big animals and 2 small animals. So he has already segregated that you are getting it as a clusters. So from the given data, how many varieties of common features are present? That will be given or coming as a clusters or association discovery between the parameters is nothing but your unsupervised learning. You are not going to define any features in unsupervised learning. You just give the data, and data itself, the program itself understand the variations between the data and it gives as a clusters. And you're not going to get any output which is a dog or which is a cat. You have to understand there are 2 varieties of animals are present. They are having different features. You have to decide which feature is corresponding to dog and which feature is corresponding to the cat. So you can see over here there are set of molecules has been screened for various descriptors. Like the descriptors means properties log p or maybe pKa values or something like similarities, Wiener indexes for the molecules. So when you are having any number of molecules with you, when you use this unsupervised learning, it try to segregate your data set into 3 different varieties of the clusters. So by this you can understand there are 100 or 200 molecules which are having same type of log p, and another set of molecules which are having same type of Wiener index. So you can work it on that one. What are the features? Further you can process that one. That is the unsupervised learning. So these are descriptions just I kept over here because majorly supervised learning and unsupervised learning are the classifications. Again, in supervised learning, you have linear regression, single linear regression, multiple linear regression, k-nearest, Naive Bayes. So everything has its own protocol to understand the features. Similarly, in unsupervised learning, you have a clustering, then PCA, independent common analysis. So ultimately, when you are using these 4 methods, your data will be coming as a clusters. When you are using these 5 methods in supervised learning, your data will be coming as a output which you have defined in the initial stage. So next important thing was machine learning applications in drug discovery pipeline and their required data characteristics. This is very interesting actually. So whenever we do the drug discovery process, our process starts from the target identification and validation. So if you want to do target identification and validation, that means whatever the protein you are selecting for the disease condition, whether that protein is targetable or not, whether it is good for the pathogenesis of the organism or not. So like that, you are going to do the identification of the proteins and validation. In that, what you have to do was, you have to prioritize the targets based upon the gene disease association. So you have like COX2 proteins and P53 proteins in cancer. Okay, the people will give more preference to the COX2 protein to target instead of P53. Why? Because P53 was generally present in normal cell and cancerous cell, and phosphorylated P53 is having more effect on the cancer, not the simple P53. But when you take COX2 protein, it was majorly overexpressed in cancerous tissues. So you have to understand which gene is more important for the disease condition. Then similarly, you have to understand what is the druggability predictions. This can be done by doing the experiments or by in silico methods. And also you have to understand what are the alternative targets. These are the 3 important data we require if you want to validate your particular protein. So in order to understand these things, if you want to implement machine learning, you require data from the successful and failed clinical trials. Then only you can design or you can identify which target is very important. But getting successful clinical trial data and failed clinical trial data is not so easy, because most of the clinical trial data will be stored at clinicaltrials.org, but this is not open for all the people. The next important thing was high dimensional target disease drug association data sets. So people may be doing some in vitro studies, or during the clinical trials, some patients may be dying, but the data from the patients who are dying from because of the clinical trials is not available. Okay, getting that data is very difficult. Then data from disease and normal state, because this is very important. The cancer which is present in Indian population and American population was different. The CT scans which we take here and some other country was different. By using different types of machines also gives different types of parameters. So getting a universal type of parameters for the disease state as well as normal state is the big task when you want to implement the machine learning programs to understand the data connectivity. So if you could able to get this data correctly, you can implement machine learning, and directly you can say that like the P53 was the maybe more effective in American population, not in the Indian population. Like that, the connectivity you can easily understand. The next method was the next step was the compound screening or lead discovery, where you have to design molecules with a desirable properties so that it can show more potent activity, and that molecule has to be synthesized at a lower cost. Why? Because when we use a lot of computational tools for designing a molecule, we will come up with a new molecules, but the synthesis of that particular molecule is becoming a problem. It may be taking more steps, or it may be involving more amount when you go for a large scale. So if these things are available, then you can easily generate new molecules. So if you want to do compound screening and lead discovery by using machine learning, you require small molecule data. So here the small molecule data is nothing but there are a lot of databases are available in the literature, like your drug bank, zinc bank database, EMBL database, SNX database, eMolecules database. So the people already generated the databases where the small molecule structures, their biological activity, and some of the properties already deposited in the databases. So you have to just go to the database and download the molecule sets and implement machine learning models to understand the connectivity between the IC50 and various properties. Then the compound reaction space and rules. So if you are designing a new molecule, what are the off targets for that particular protein or for that particular small molecule? What is the metabolism? What is the toxicity? So all those things you have to understand. Then similarly, if you want to do the any type of binding affinity studies with a small molecule to the protein structure, you require a 3D crystal coordinates of the protein structure. So these are the data required if you want to do machine learning implementation at the stage of compound screening and lead discovery. The next step was preclinical development. So in preclinical development, we are going to understand what is the biomarkers available and what is the drug response signatures and prediction of biomarkers of clinical endpoints. These are the three requirements you are going to address. So you require the data which is related to biomarkers and protein connectivity. So protein connectivity is nothing but what are the like different pathways by which cancer is going to be emerging, what are the proteins which are involving over there. So if you could able to have a data which is related to biomarkers and different proteins which are involving in the disease condition, you can implement machine learning models over there. The next stage was clinical development. Here we are going to determine the drug response by cellular phenotyping in oncology and precise measurements of the tumor microenvironment in immuno-oncology. So you require the data, available clinical trial data, which can differentiate cancerous cells and normal cells. Like most of the time clinical trials data is available, they try to give the information about the cancerous cells, whether the cancer condition was increased or decreased and what are the upstream conditions which have been observed in a patients. But none of the data is available with related to the normal cells, what happening to the normal parameters. Then you require high number of images for clinical trial. So most of the time, because of the cost, people will take 1 or 2 images per day, until unless you get more number of the images from same patient or from different patients from different origins, then you could able to understand what is the connectivity between the like size and weight of the tumor with respect to the age, maybe sex, or it may be some other parameters which are present in the patient. So here similarly, when you are understanding the connectivity between the cancer cells and normal cells, you also require protein connectivity. So these are the various features where you can implement machine learning. So this is a nice picture I have taken from the recent publication, Nature Reviews Drug Discovery. So as I said, you like in supervised techniques, you have a lot of methods are available. Then in unsupervised learning, you have a lot of statistical methods are available. So as I discussed in the previous class, people already studied all these parameters like compound bioactivity and assay readouts from virtual drug target schemes, as well as novel therapeutic targets from target gene association. So association between the protein and the gene has been already studied by the machine learning. Similarly, the druggability and the pharmacokinetic properties, how it is related to the protein structure or a sequence. Then de novo molecule design. If somebody wants to design a new molecules, they have already used unsupervised learning technique and designed the molecules. Similarly, cancer related genes from the RNAi screens, drug sensitivity prediction, ADME properties and chemical synthesis, QSAR, then docking studies and dynamic studies for the molecules with particular proteins, and image based diagnosis, MRI data acquisition, all these already done and which are related to various stages of the drug discovery by various scientific groups. You can just go through this paper, and once you go to this particular reference, you can able to see what is the method they have implemented and what is the output. Here there are any type of method you can utilize. So I think it is visible now. You can implement based upon the data, either supervised learning or unsupervised learning. Could able to write a program to understand what are the properties or the connectivity has been already designed. So coming to the machine learning role in healthcare. So as I said you, in different stages of the drug discovery, we have a programming already available, and people are still working on that to curate the data as well as to get more connectivity results. So in case of disease identification and diagnosis, we have a company from USA, Quantex, is working on disease identification and diagnosis, and they could able to give the connectivity in a Parkinson's disease in different parameters of the patients, and they could able to treat the patient accordingly by using and diagnosing the stages of the Parkinson. And similarly in drug discovery and manufacturing, InSintro was the company which was by the Microsoft. They are working tremendously to bring new molecules by using machine learning methods, by understanding the connectivity between the structure, then their activity, as well as various descriptors of the molecules. The next important area was medical imaging, where the Microsoft Inner Eye company was working on that one. Understanding the images produced by various machines and the parameters, based upon that trying to get a connectivity between the image sizes and normal tissues and cancerous tissues. If the same benign and malignant tumors are present in a brain as well as in some other tissue, what is the connectivity and how they are similar. So all those medical imaging as taken by the Microsoft Inner Eye. Then coming to the personalized medicine and treatment, here the Watson Healthcare of IBM is working on personalized medicine. So here they are understanding the gene and disease condition correlation, based upon based on that trying to treat the patients. Similarly, Seox from a European health company working on the smart health records, like it's almost equals to, you might have listened, one of the Indian company, we are working on a smart idea, like where the TB patient is taking the tablet in every day or not. So such type of health records they are maintaining, so that whenever the patient is having a problem, if he is forgetting to take a medicine, he will get a alarm that you have forgotten the tablet today, so please take, like that the messages are going to the patients. This will be a smart health record where the machine learning models has been implemented. Then the next method was the disease prediction, where the researchers at the University of Nottingham in the UK are working to understand what are the, if any person is having like diabetes and obesity, what is the probability of getting a heart disease to that person? What is the probability of getting a Parkinson's to that person? So based upon the already existing health records of the patients, machine learning models are used to understand what is the probability of getting a new disease and at which stage is present to get that new disease condition, or they are going to give you what is the like disorders he can experience, so that prior prediction gives them to what is the level of sugars he has to maintain and what is the BP levels he has to maintain. So these are the different machine learning applications in healthcare. So if you want to write a machine learning program, what you require, what knowledge you require? As I said, you need to understand the biology of the molecule or the biology of the protein or the disease condition, as well as you have to understand what is the chemistry behind that interactions, protein drug interactions, and how these interactions can be studied either by using structure based drug design or ligand based drug design. Then how we are going to represent this data? I know that my ligand like celecoxib is binding to the COX-2 protein, but how I can represent to others? What is the parameters? There you are going to see like hydrogen bond interactions, hydrophobic interactions. But when you are using a machine learning for large set of molecules, the images for showing all molecules is not so easy. So when you are handling a larger data, you have to represent those in the form of graphs or in the form of clusters, as we have seen in unsupervised learning. Segregate your molecules which are having hydrogen bond interactions with particular protein and hydrophobic interactions with particular amino acid. So you require some of the programming knowledge if you want to handle large data. So those are nothing but Python. You have R, SQL also available, but nowadays all the software companies are moving towards the Python because it is user friendly when compared to the other languages. Then if you are doing some statistical calculation like simple linear regression, multiple linear regression, if you want to use by programming language, you have to call those two packages, NumPy and Pandas. NumPy is a programming language in the Python. This is basically applicable when you want to do MLR or simple mean median mode calculations in a Python. You have to call that particular program. Then only you could able to do the calculations. Similarly, pandas are nothing but this is a package which is used to use the data which is given in the form of tables or the Excel sheet. When you have single molecule or single number, you can use directly by Python. But if you have large number of data, as I said you, when you have a clinical trials, you are going to generate multiple data which are related to normal patients and cancerous patients in different days. So all the data will be converted into the Excel sheet, and that Excel sheet data will be used by using Python programming to do the SLR methods. Next program was Matplot library and Seaborn. These are the two graphical representation packages. Whatever you are doing in an Excel sheet, generally when you do concentration versus assay activity, you will plot a graph, x-axis versus y-axis. So, but here you are having multiple parameters. The cancerous, for example, if you are taking about the cancerous patient, the BP and the cancerous tissue levels and the proteins which are involving in the cancerous tissue, there are multiple parameters are there. So when you want to plot a graph between multiple dependent variables and independent variables, you are going to use Matplot library and Seaborn. These are the two packages by which you can write a Python programming and you can represent your data. So now coming to the important packages, RDKit and ChemPy. These are very important for a chemist. Numbers and the text you can easily understand, but the molecular structure, two-dimensional structure and three-dimensional structure, and what is the log P, what is the molecular weight of those molecules. So all those things, if you want to calculate, you have to call these two programs. One is RDKit and ChemPy. RDKit basically for the chem informatics calculations and ChemPy for the reaction kinetics. So all these four will be short pieces for you for doing a machine learning programming. So basic machine learning packages are scikit-learn and TensorFlow. These are the two packages. Whenever you want to handle bigger data and try to get a connectivity, you have to go to scikit-learn and TensorFlow packages and write your programming by using all these four different varieties of packages. Okay. Then you have Keras and Spark. These are the two packages. These are used for the deep learning. So scikit-learn and TensorFlow can handle maybe 1000 to 2000 molecules or maybe up to 5000 set of data molecules. But Keras and Spark can handle unlimited number. So whenever you want to do higher end data, you are going to use the deep learning method, that is by Keras and Spark. So why I kept all these things here means, though you are a pharmacist or a chemist, if you want to grow in the area of machine learning, you have to learn these packages. It doesn't require much work. If you know the logic, how I have to calculate the log P of a molecule, how I have to calculate the molecular weight of a molecule. So molecular weight can be calculated by counting the molecular mass of the carbon, hydrogen, oxygen, and nitrogen. That's simply you know. So you write a program in such a way that how many number of carbons are there in your molecule or in a Smiles notation, whichever you are giving. Based upon that, for example, if you have 6 carbons in your Smiles notation, 6 into 12 will become your molecular weight of the carbons corresponding. So like that, you are going to write a simple programming by using RDKit. Uh, Durgesh, how much time I have?
Yes, ma'am. Still, we have 10 to 15 minutes. You can proceed, ma'am. No problem.
Okay. So just I want to give little demo about the.
Yeah, yeah. Sure, ma'am. Sure. No problem.
So let me show you how the molecule can be represented by simple programming. So RDKit, as I tell you, it is a package for the chem informatics. Whenever you want to write a program in the RDKit, let me show you visualization, like how you are going to import a molecule. You don't have in ChemDraw or Marvin view with you, but still you want to draw the molecule. Okay, how you are going to draw it and visualize it. So let me show you directly in the RDKit. For doing programming, you require either Python or Anaconda or the Jupyter, the Python environment to write the programming. So I'm going to import the RDKit package. It is already available in the environment. So I'm going to import RDKit. From RDKit, there are sub packages are available, like chemistry package, drawing package, IPython console. Whatever you are drawing, that will be converted into the Python. Then descriptors like 1 dimension, 2 dimension, 3 dimension descriptors. If you want to calculate, you have to call this particular package. Then all chem, so all chemistry related calculations. Then data structures, whatever you are giving in as an input, that will be converted into the data structures. And next important thing was the numpy as np. So if at all you are doing any statistical calculations, you have to call this program. It's very simple. You are going to call the RDKit and various packages and trying to write your Smiles notation. Here I have just written molecule chem dot molecule from Smiles, and this is the Smiles notation of your paracetamol. Let me show you. So you just write this one and click over there. You are going to get a structure of your molecule. So this already I have performed. So this will be your input. You are giving the Smiles notation to the RDKit, and that Smiles notation is going to give you the molecular 2-dimensional structure of your Smiles notation. So similarly, if I want to calculate total polar surface area of the molecule, I'm going to write a simple program like from RDKit, import the descriptors and give your Smiles notation of the molecule and write your total polar surface area. Whichever you want to calculate, you just write over there. So let's see. You can see I'm just calling the total polar surface area. I have given the Smiles notation of my paracetamol molecule. The surface area was 49.33. Similarly, if I want to calculate log P, hydrogen bond donors, acceptors, molecular weight, rotatable bonds, which is nothing but your Lipinski rule of 5. So you just give the Smiles notation of your molecule and it is going to give you what is the log P, donors, acceptors, molecular weight, and rotatable bonds. So for doing this, you don't require ChemDraw or Mol Inspiration software or any software to draw the structure and calculate the Lipinski properties. Directly, simple writing the script in Python, you can able to calculate the descriptors. And this Python environment and RDKit are free, so that you need not to pay anything for the calculations. So similarly, if you want to calculate the gas tiger charges, the charges which are present in your molecule, you are going to write a simple program which is around 5 lines. Okay. So I have performed this one. So whenever you are performing any type of calculation, you have to don't forget to call your RDKit. First, you have to create your environment as a RDKit, then only you can able to do the calculations. So by writing this simple programming for the paracetamol, you can see the charge distribution is basically present in these locations wherever the highlighting of the colors has been done. This color highlighting also, this is user friendly. You can change the colors depending upon your need. So, but to do this calculation, you should know what is the programming script. So for example, here I'm following a certain pattern, like the smiles notation is going to be shown in a parenthesis. Whether this parenthesis is to be how big, how it is going to be closed, whether it is a square brackets or normal brackets or simple parenthesis, where I have to put the colons. All those things will be learned when you work on a programming. The fundamental rules of programming you have to learn if you want to write a script. So this is about RDKit. Now just I want to show you simple machine learning programming by using scikit-learn. So let me show you. So you can see I'm just first calling all the RDKit packages, because here this particular program has been written to understand whether the given molecule is active or inactive. So as I said you, when you are doing the supervised learning, first you have to define your inputs and the features. So here I am defining 4 molecules, you can see, with 4 different smiles notations. And in this, I'm going to divide first 2 molecules are active molecules and second 2 molecules are inactive molecules. I have defined over here based upon the fingerprints. So it's very important to know how to represent your smiles notation and generating the fingerprints. Fingerprints are nothing but zeros and ones conversion for your molecule. Once this has been done, it is going to generate a vector that is nothing but your the connectivity between the similarities. Then I'm using random forest classifier, that is a supervised learning method, because there is no linear relation. So
Hello.
I have used, hello.
Excuse me, ma'am.
Yeah.
Whatever the packages you are showing on the online streaming, it is not visible. The only PowerPoint presentation is visible on this.
Oh, sorry. So I think this is visible for you people for writing a small molecule.
Yes, ma'am. Now it's visible.
Okay. So you have to import the RDKit. So whatever the molecule you want to convert into the 2-dimensional structure, you give a smiles notation, and it will be given as the output. You can see this is the output which is coming in the 2-dimensional structure. So when you want to get this structure, you have to call the main program RDKit and the sub packages like chemistry. So you have given a smiles notation, but it has to be drawn. So you are going to call the program drawn. So similarly, next method was to calculate the descriptors. So I have given a paracetamol molecule. In order to get a total polar surface area, I have to write only 2 lines program, descriptors.tpsa, where it is going to give the output as a 49.33. Similarly, if you want to calculate Lipinski rule of 5 for your molecule, give the smiles notation and ask for the calculate descriptors. And these descriptors, whichever I want to calculate, I have defined over here: molecular log P, donors and acceptors, molecular weight, and rotatable bonds. So you could able to get the output in a fraction of seconds. You don't require mol inspiration or any type of the package to calculate the properties. Not only these Lipinski properties, any higher end properties like 3-dimensional descriptors or 2-dimensional descriptors, or it may be electrostatic package over the molecule, you can directly get from the RDKit. So one example I have shown over here, if you want to understand what is the charge distribution on your paracetamol molecule, you are going to write simple programming, which is of 5 lines, which is going to calculate the gas tiger charge distribution on your molecule. And the colors can be varied depending upon your need, whichever your properties you are defining in the programming. Based on that, you are going to get the colors. So next important thing was the machine learning programming. I want to just understand from a given set of molecules, understand the fingerprints, and try to, if I give a new test molecule, whether it is active or inactive, based upon the fingerprints it has to generate. So here I'm just taken 4 molecules for the study purpose, and these 4 molecules I asked to convert into a fingerprints. Fingerprints are nothing but zeros and conversion of bits. So once that is done, the connectivity between the fingerprints will be identified by using the vector formation. And here we are going to use the random forest classifier supervised learning method. And I segregated the 4 molecules into actives and inactives. First 2 are actives, where I am representing it as a 1. You can see I have written small text over here with the first 2 molecules being actives and the last 2 molecules being the inactives. I have defined that one for understanding purpose, because as it is a supervised learning, I have defined the inputs as well as I am giving the features, calculate the fingerprints, and divide my molecules into actives and inactives. Okay. So feature conversion as well as the input has been done. Now we are going to give a new molecule, which is new Smiles notation you can see when compared to the above 4. So what is the nature of this particular molecule, whether it is active or inactive, depending upon the fingerprint similarity. So it's going to give me output as a 1. 1 is nothing but active. As I said you, I have defined the first 1 corresponds to active molecules and 0 corresponds to inactive molecules. So whatever the new molecule I have defined over here, the program can able to tell it is an active molecule based upon the fingerprint similarity, which is already calculated by using training set. This is simple machine learning programming, how we are going to write. If you understand carefully, this is the RD kit which is related to define the smiles and calculate the fingerprints. Then you have a numpy which I am using to convert into the array form, like whatever the zeros and ones fingerprints I am getting, that I am converting into the matrix form. And that matrix, once it is done, I'm using sklearn, that is nothing but scikit-learn, which I told you. If you want to implement MLR method or supervised, unsupervised, or reinforced methods, you have already existing background packages are available. sklearn I'm using to use the random forest classifier. So this is the only, see, if I want to write multiple linear regression, I have to write almost 50 lines programming. Like first calculate the slope of the y-axis and x-axis and the coordinates and the descriptors. You have like 50 lines program. Instead of that, if you use sklearn, it's already inbuilt with this particular name. So random forest classifier is having the already written code, which is in the form of ensemble. So here just simply you write random forest classifier so that it will calculate the background information which is required for the particular type of statistical calculation from the given input data. So after that, you are going to just make a fitting graph between the fingerprints. Then again, you are using the text molecule, which is in the form of smiles notation, and getting the output. So if you want to write any machine learning program, you should understand what are the different packages are available, how we are going to represent your small molecules by using RDKit or ChemPy. And if you want to generate or convert those molecules into a graphical representation, that is NumPy, as I told you, or Matplot library. So all these are combinations. The only program which you are using machine learning was simple 2 to 3 lines, which is a package for the statistical calculation. So for understanding what is the nature of this molecule depending upon the fingerprint similarity, I don't require any software. Simply I can modify this smiles notation. I can calculate for any molecule whether it will be active or inactive based upon the fingerprint similarity. So this is about machine learning programming. If you understand how to represent your different packages in it is program Python, you can able to write your machine learning programming. So coming to the machine learning startups and platforms which are available. So recent survey from Bench Science blog, it was showing that 230 startups are using artificial intelligence in drug discovery. It's just startups, but established companies like Pfizer, Reddy's, and AstraZeneca already tying up with IBM to do the drug discovery by using machine learning models. And this is one of the company which is present in South Korea, working on disease target relationship.
Hello, ma'am.
Yeah.
Hello.
Hello.
Ma'am, presentation screen to be shared.
Yes. So 230 startup companies are working on artificial intelligence, and Aggregate and Synthesize is one of the company from South Korea which is working on discovering the relationship between diseases, targets, and drugs. It's basically helpful to repurpose or repurpose the already existing molecules. That was the motto to start to understand the relation between the already existing diseases and targets. Like that, you have 230 companies which are working in various levels of the drug discovery process. So as I said, you can do the machine learning by using scikit-learn or TensorFlow. But if you are handling very high data, in that case, you can use the platforms which are available from Amazon, AI1, Apache, IBM Watson, and Microsoft and Water. These are the various platforms which are available where you can do handling high-level data. So this is about the brief introduction about the machine learning. There are complicated programming you have to write, but I have shown very simple programming to understand. Once you are familiar with the programming, you can play with it. But when you want to write a Python or machine learning programming, you understand what is the logic behind that study. So once you understand logic, then only you can implement all the programming skills. So you require subject of your chemistry as well as programming and understanding the relationship between the biology and chemistry in order to design any logistic relationship between these various subjects. So with this, I want to conclude stating that drug designing is an art, but the application of informatic resources and data analytics will play a major role in the attempts to make it more rational and successful in the future. Thank you.
Hello.
Hello.
Thank you, ma'am, for such a nice and wonderful informative session. Especially, it is very useful to the PG students, researcher, as well as the academicians. And I hope all the participants understood the concepts of machine learning platform in the drug discovery process.
Thank
So
you.
now proceeding towards the question hour session,
Yes.
I would like to request all the participants, those who want to ask any questions regarding this. Yes,
Those
those
participants who
raise
want
your hand.
to ask the question can raise your hand
Please
so that
raise
we
your
can
hands.
unmute you.
Hello.
Hello.
Yes, Mahabar ma'am.
Sir, there is one question coming from Mrs. Ketaki Dhane madam
Okay.
to Mallika madam. Ma'am, is it there any free software available for this type of calculations? Because as an academician, it's a bit difficult to get all purchase software and also she's
Yes.
asking about any free software available for this.
Yeah. I can understand as an academician there are constraints for the money, but
Yeah.
whatever I have explained over here, all are free softwares.
Okay, that's
RDKit, Python, Matplot library, scikit-learn, all are free packages.
Okay. And second question from her only that what is the percentage of acceptance, that is whatever machine learning calculations are there and whatever in vivo correlations we are getting, is there any data available regarding the percentage acceptance?
Yeah, it is almost 60% acceptance is there. Why? Because here, whatever the machine learning models we are using, we are training. So
Yeah.
how best you train your data, the output is going to be getting that much best. So if you are training the data in a improper correlation or the improper logic, then the data output is going to be improper. So as a programmer, as a chemist, you have to decide what are the properties you have to correlate. Based upon that, the success rate depends.
Okay.
Hello, ma'am. One more question is there from Chenchu Lakshmi, ma'am.
Okay.
Can we provide training to the students by using this machine learning tool?
Yeah. Yeah, the
She's
motto.
asking regarding Miss.
Yes.
Can we provide the training to the students, especially to the
Yes.
PG students?
Yes, yes. The motto of starting Mass Training Academy was the same thing, because I worked in a Naipur, Hyderabad for almost 7 years. But I can understand the PG students are more concentrating on the synthesis, not on other parts. But when you go to the industry, the query comes, what you do with the synthesized molecules, whether it is a active molecule or not. So in order to give like interest in all the type of areas, I started Mass Training Academy, where I'm giving training for the CAD tools. I think Rashmi already finished the CAD course.
Yes.
Yeah. Yeah.
Then
Yeah,
I'm,
madam.
yeah, and also I'm giving Python and machine learning course.
Yeah. Yes. Any more questions from the participants? I would like to request those who are means those who want to ask any questions, queries regarding this session. Anyone?
There was some query I can see over here.
Uh, yes, ma'am.
Can you please provide us a link for those softwares?
Yes, yes.
Yes,
Some
yes.
participants are asking regarding the that presentation.
Okay.
Yes, ma'am.
Yeah, links actually, whatever I have written over here, you just type over there in the Google like Python.
Yeah.
You will be directed towards that particular website. There is
Okay.
no extra like efforts you have to put over there.
Yeah, yeah.
Hello, ma'am. There is a one question from YouTube. Mr. Nishant Joshi was asking that how to use the drug ability score in publications.
Drug ability score.
Yes.
Drugability score. Okay.
Yes.
Yes, yes. The drugability score, you have to define in such a way that when you do the identification of the protein pockets, it is going to give you varieties of pockets which are present in the protein structure. During the publication point of view, you have to define what is the drugability score which you are observing by using the software, as well as the what is the hydrophobicity, hydrophilicity ratio, and the important amino acids. So based upon that, you have to give the interpretation for your results, and you can publish over there. No issue.
Okay. Ma'am, one more question is there. Ramalinga Shwarao is there. Kashula. They are asking, is there any training institute in Hyderabad
Regarding
regarding that softwares and other things?
like
Miss, you are
mass.
belong to ma'am that research institute is there. Na, you are particular as a trainer.
Yeah, yeah.
So they are asking about the training institute.
Okay, I myself giving the training in Python and machine learning.
Yes, ma'am.
Still, now the batches are going on. Recently, I have just started CAD programming. By another 10 days, we are going to start next batch for the Python as well as these packages.
One more question, ma'am. Just Noshin Sultan. Just a minute, ma'am. I'm just unmuting him.
Okay.
No. No.
Ma'am, if you address Mass Academy on the chat.
Your voice is not clear.
Hello.
Hello.
Nosin Sultana, ma'am. Yes, please proceed for your question, ma'am. Please.
Okay. Ma'am, I need to ask whether we have to learn first program learning in order to give program in a software.
Yeah, I can say that when you start learning programming, simultaneously you can do the
Yes, ma'am.
execution of that programs. It doesn't require like you have to study theory first, then do the practicals. Simultaneously, you are going
Yes.
to do the programming. Like Python, when you start working, when you write a script for the Python, you have to execute that one. So once you start executing the program simultaneously, you will be getting interest to do more. So it doesn't require prior
Okay.
knowledge. Directly, you can do
Okay.
the coding.
Okay. Thank you, ma'am.
Yeah.
Ma'am, one more question is there from YouTube.
Mhm.
Raji Lakshmi ma'am is there. She's asking, how much does it cost for Python or CAD?
I can share you privately because this is not platform to discuss about those things.
Yeah, yeah. Okay,
I
ma'am.
can share to Rashmi or Dr. Vijay Jagtap. They
Yes,
can
sir.
communicate to you.
Yeah, yeah. Sure, ma'am. Sure. Thank you. One more question is there. Just ma'am
Yeah.
unmuting her. Uh, hello,
Hello.
Ketki ma'am. Please ask your question. Hello, hello.
Hello.
Ketki Dhane ma'am speaking.
Uh, yeah.
Please ask your question to ma'am.
This is
Please.
Malika. This is Malika, ma'am.
Sorry. Yes, ma'am. Please.
Ketki
Uh,
ma'am, there is a disturbance from your side.
Ketki Swapnil Dhane ma'am.
yes,
Hello.
sir.
Please, please ask your question. Your voice is not clear, ma'am.
I think the query was, can you provide the online training
Online
for
training,
those?
for
Yeah,
yes,
yeah.
yes.
Yes,
She
yes.
has asked.
Yeah, yeah. We will provide the training. I'll share the details
Ma'am,
to the
one more is there.
programmers.
Miss Amruta Shinde is.
Mhm.
Amruta ma'am, can you ask your question?
Taking isolated compounds, any purity like that. That is a purity we need to check by using. But there are so many methods to check the purity. It starts from DNC, then even you can do HPTLC is mainly
Gautam sir, have you unmuted all?
of the compound.
Yes, yes.
But if it is more than
Principal sir said that
99.99%
unmute to all.
No, there
pure, I
is
think
too
that
much
will be
disturbance.
sufficient to carry out any kind of biological study. Normally, if the isolated compound is totally novel, for example, if it is now not novel compounds, maybe you can compare with the standard
Sir,
purity.
mute all.
Mute
If it is
all.
a novel, totally new and original compound,
Because someone is
isolated
talking
from that
somewhere
particular
else.
medicinal plant means what you need to do. If it is more
Hello, sorry ma'am for the disturbance.
No issue.
Okay. Sir, we can take the question one by one because by unmuting all, it will create too much disturbance.
Disturbance. Okay.
I think there is no question from chat box also.
One more question is there. One more question, ma'am. From Omkar Pendse. Can the Mars Academy provide the distance learning?
Yeah, it's online session only. We are
Okay.
providing.
Yes.
All are online training programs.
Okay.
Okay, I think.
Sir, yes. There is no
yes.
further one more questions are there.
Okay.
Sir, shall we
Just a
proceed further?
Yes.
Yes, please, no problem.
Yes, sir. I would like to request Mahaba ma'am, Rashmi Mahabar ma'am, session coordinator. Please proceed further.
Hello.
Yes, ma'am. Proceed.
Uh, I would
Yes.
like to thank Dr. Mal for sharing such a nice as well as the information because chem informatics or machine learning is considered as good as the tip of iceberg. As you go deep, it will be a sea for you. So definitely, ma'am, this is a good platform which is shared by you, so that the participant session will definitely get benefited, and whatever hidden areas or machine learning or deep learning, these are the recently introduced areas wherein the work is still going on. So definitely the participants or researchers, PG students, those who are going work on this, definitely have a new focus on their research studies. And with your guidance, with your help, definitely they can improve their knowledge, they can give something new to the world. Once again, I would like to thank Dr. Mallika Madam on behalf of Yeshwantrao Bhosle College of Pharmacy team for being part of this particular webinar series. And definitely, we are also expecting from you, ma'am. Definitely.
Thank you. Thank you for like giving me opportunity to discuss with the students and faculty from various areas. I'll thank Rashmi as well as Vijay and Durgesh providing me platform.
Thank you, ma'am. Thank you so much.
So all the details which are related to the queries, I will share with you, any one of you, so that you can share to the participants.
Yeah, sure, ma'am. Sure.
Thank you.
Now I would like to request all the participants, please share your videos for clicking a one photographs, please. Start your videos. Yes.
Sir, it is giving a message that host has disabled. Please enable it.
Okay, just a minute. Rukai sir. Hello.
Hello.
Rukari sir.
Sir.
Can all the participants share their videos right now?
Okay, we will just unmute all of them.
Unmute, no? Okay.
Hello, sir. Done it?
Have you captured, sir?
I think Jacob sir is
capturing the snapshots.
Yeah, I will also do that.
Yes, sir.
Yeah, done from my side.
Okay, sir. Thank you, sir.
Okay, sir. Yes, yes. Thank you. Okay, all the participants can leave the.
Yes, thank you so much.
So thank you so much.
On the behalf, yes, on the behalf of Yeshwantrao Bhosle, you have patiently listening throughout the whole session, and thank you so much. Thank you to all the participants throughout the country and the state of Maharashtra and different parts of the country. Thank you.
Thank you, Mallika ma'am. Thank you very much. Sir, close the meeting now, and it will proceed for the saving the video.
Saving. Okay, just end now.
Hmm.
Shall I proceed for end?
End meeting for all.
Okay.
And it will